Now, most of any biological system is mostly going to be largely comprised of water. Okay, so that means this is the main solvent for any biological system, so inside the cell or otherwise, right? And so the polarity, so the permanent dipole of the water, is really important on influencing what types of molecules are soluble, right? So let's go back and look at our example of sodium chloride, right? So let's say we have a sodium cation, and then we know that table salt, sodium chloride, is very soluble in water. And so one of the reasons for that is because of the permanent dipole nature of the water. So the water, because of its dipole, can interact with either the sodium or the chloride anion, depending on how the water orients with these two molecules. So because the sodium ion is positively charged, the negative end of the water will interact with it. So for instance, like this. Okay, whereas the chloride anion is going to be the opposite. So the positive end of the water will interact with the negatively charged. Oops, other way around. So this permanent dipole is basically very versatile in how it can handle either positive or negative species. And so that just changes the orientation of how the water interacts. But importantly, what it forms, this layer of water that forms around the ions, these are called hydration shells. Okay, and so for any molecule, if a sufficient hydration shell that can be made that can interact with the bulk media or the bulk water in the system, then these are going to be the easier that these hydration shells can be made, the more soluble that the molecule will be. So in this case, sodium chloride, it's very easy for the negative part of the water dipole to interact with the positively charged sodium ion, and for the positive part of the water dipole to interact with the negatively charged chloride ion. And so as we know, sodium chloride, so table salt, is very, very soluble in water. And so things that form similar interactions with water, such as hydrogen bonds or dipole interactions, are going to influence the solubility of a particular molecule. So we can also think of other things that may hydrogen bond, such as an alcohol group. So we just take a sort of generic alcohol group, right? It's going to have lone pairs, all right, which allows it to hydrogen bond with water. So they can either form, for instance, we'll draw this geometrically correct. So we can form a hydrogen bond with the water this way, where the hydrogen from our alcohol group is the H bond donor, and we'll get back. We'll go look explicitly more at H bond donors, acceptors in a second. But for right now, let's just note that this is an H bond donor. Water is acting as an acceptor. Water can also act as a donor, and the lone pairs in the oxygen can act as an H bond acceptor. So we can have another interaction like this, and then the same with the other lone pair on the other side. All right. And so this helps to create a hydration shell around the alcohol group of this molecule. And if the molecule has a sufficient number of these functional groups that can interact with the water in such a way, a hydration shell will be able to form, and that will increase the solubility of any molecule within the water. Okay.